Hello, everyone. I'm Chen Meng, a PhD candidate of the School of Biological Science and Medical Engineering at Southeast University. Thank you for attending my presentation. It's a pleasure to present our research at the conference. The primary objective of this presentation is to share our findings on the effects of nickel ions on spontaneous neocortical network activity. I will begin with an overview of the background, followed by our methodology, key results, and finally the conclusion and the implications. Nickel is widely used in implantable medical devices. Under physiological conditions, these implants undergo corrosion and release water-soluble nickel ions. Nickel ions-induced cytotoxicity remains a central focus in biomaterial biocompatibility research, with numerous studies characterizing its cellular toxicity profiles and underlying molecular mechanisms. Driven by advances in neurointerventional techniques, nickel-based alloy implants such as embolization coil, flow diverter, and aneurysm clip have increasingly been adopted in neurosurgery and are now routinely used for treating cerebral vascular malformations. Clinical studies have reported neurological deficits such as seizure, limb weakness, and language disorder in some patients treated with cerebral aneurysms using nickel-based alloy implants. Thus, elucidating the neurotoxic mechanisms of nickel ions is both scientifically imperative and clinically urgent. Nickel ions is a common T-type calcium channel and NMDA receptor blocker. It can directly and rapidly influence the electrophysiological properties of neurons. MEA technology is a well-established platform for interrogating neuronal network dynamics. An Italian group have demonstrated that 300 micromolar nickel ions can significantly reduce the spontaneous activity firing rate and burst duration of cortical neuron network cultured on MEA. However, in our prior works, we systematically characterized the concentration-dependent effects of nickel ions on spontaneous activity in neocortical networks of rat brain slices. Strikingly, even at 500 micromolar, nickel ions fail to alter the spontaneous firing rate or bursting rate. This result challenges the interpretation that nickel-mediated ion channel inhibition should suppress spontaneous network outputs. To rigorously test this discrepancy, we expanded our sample size and performed comprehensive analysis of spontaneous activity before and after 500 micromolar nickel ions exposure. In the experiment, a single neocortical slice was positioned onto the central electrode array of an MEA chip and gently secured using a stainless steel ring fitted with a fine nylon mesh. The slice was allowed to equilibrate for 45 minutes to ensure full recovery from preparation-induced stress. A baseline 45 minutes recording of spontaneous network activity was then acquired and served as the control condition. Thereafter, the perfusate was switched to oxygenated ACSF containing 500 micromolar nickel chloride, and after a 20 minutes incubation, a second 45 minutes recording was obtained from the same slice under nickel exposure and served as the experimental condition. In data analysis, we assessed the mean firing rate of spikes and the mean bursting rate, which are the two basic and most commonly used measures in MEA-based neurotoxicity test. Additionally, 4 established burst-related metrics, the percentage of spikes occurring within burst, mean burst duration, mean number of spikes per burst, and the mean firing rate in burst were quantified. Changes in these parameters before and after nickel exposure were investigated to assess the impact of nickel ions on neocortical neuronal network spontaneous activity. The results indicate that nickel ions exert no statistically significant effect on either the mean firing rate or the mean bursting rate, which is consistent with our prior findings. However, nickel ions reduce burst duration, which is consistent with the Italian group's report mentioned above. Nickel ions also induce a reduction of the percentage of spikes in burst and the mean number of spikes in burst. These alternations reflect both a disruption of the network's intrinsic bursting architecture and a more temporarily dispersed pattern of spontaneous activity. Neocortical networks in brain slices containing a defined proportion of inhibitory neurons. These inhibitory neurons could play a dominant regulatory influence on spontaneous firing rate compared with excitatory neurons. Consequently, reduced inhibitory neuron activity or compromised inhibitory synaptic transmission would be expected to disinhibit network activity and elevate overall spontaneous firing. Concurrently, nickel ions also suppress intrinsic excitability at the single neural level. We propose that these opposing effects, a network-level disinhibition and a cellular-level inhibition, counterbalance each other, explaining why mean firing rate and bursting rate remain unchanged following 500 micromolar nickel ions exposure. This research yields three key implications for MEA-based neurotoxicity testing. First, spontaneous firing rate is not always a reliable indicator of changes in neuronal network excitability. Second, inhibitory neurons may exert substantial modulatory influence on spontaneous activity. And third, in vitro cultured neuronal networks can exhibit pharmacological response profiles distinct from those observed in acute brain slices, owing to differences in cellular composition and synaptic connectivity. These findings provided valuable methodological guidance for the future development of MEA-based neurotoxicity assays. Thank you for your attention. I will be happy to answer any questions you may have.